So I wanted to pick up a little bit on something I threw out yesterday as kind of a teaser at the end of my talk when I put the DNAnexus clinical data platform up onto the slide deck. We have been working with a major pharmaceutical company to bring our quality, basically to get a quality management system implemented in order to be able to incorporate DNAnexus as a platform inside clinical trials. We're finding more and more use of NGS in clinical trials, and we really want to be in that position. I mean, it's still early days, but more and more we're seeing NGS technology being used in the clinical trial setting. Let me see if I can get this thing working. I'm not used to this type of thing, but now I think I'm on the right slide with the one on there, and I'm looking at it correctly. So, the current state of clinical development really is a case of much data, but very difficult to tease inside out. Trials, as anyone in here who's in the pharmaceutical and drug development knows, it's amazingly expensive. It's eye-wateringly expensive. $2.6 billion per drug, 8.5% over inflation. And the protocols, I thought this was pretty funny when you're up to 169 from 55 in a decade, the pages per protocol, just for a case report form. It's also quite fragmented. A single trial can run 725 different systems. There's hundreds of different vendors serving in this space, and it really cries out for some uniformity. We're seeing a definite rise in the use of NGS in clinical trials. The precision medicine and genomic technologies, I mean, really, we have to have these new trial methodologies emerge. There has to be. The other good news is that the FDA is embracing new trial methodologies. The new commissioner has been very, very clear about his desire to look at alternative ways to move drugs through discovery and into clinical use. As we see genetics come into play and genomics, we can see more and more targeted therapies, more companion diagnostics. There's a number of different areas where we can use NGS in the clinical trial context. We think that it's likely that the clinical trials using NGS will be able to reduce time, reduce cost, and I think most importantly, increase safety and efficacy by being able to target drugs to people's whose genetic makeup we know will be amenable to the drug, or the drug will actually work for them. And biomarkers in particular, when I was at AGBT Precision Health last year, it was very clear that by having a biomarker, you increased your chances of getting out of phase 3 by 3x. 300, you'd be 3 times as likely to come out to an approved drug if you had a biomarker going along with it. It's not a silver bullet. It's pretty tough to come up with biomarkers, and genetic biomarkers that are related to a single nucleotide polymorphism, they're few and far between. It gets to be a much more complex and nuanced picture. Clinical drug development can be reduced probably from 1.8 to 1.4 billion. If you have a biomarker, that's a 26% savings right there. The trial success rate goes up from 8 to 26%, which is really a big deal. That gets you to the point where you're almost getting a third of your approvals for your trials. And of course, faster trial completion. The amount of money that's spent on these trials indicates that even if you could shave a week or 2 weeks off of the trial setup, if you could get your answers even a bit faster about safety and efficacy, basically be able to look down the road using genomics, and it's absolutely clear that having biomarkers can get you to a faster trial completion. In the oncology space, you can really see this as well. 12 to 15 years and $1 billion to get an oncology drug to market. 70% fail phase 2, about 60% fail in phase 3. And if you were to take a look at a specific example where we had a biomarker for HER2, HER2, you have a success rate increase of 23%. And I think most important, you've got well, not most important, the success rate is important, but having the cost drop down by 27% is a really big deal. Again, biomarkers and other things are going to be very useful in the clinical trial context. So I wanted to go over this diagram again, just a little briefly, because we did cover this a bit yesterday. What we realized is if we could have the right level of security and compliance, namely have a quality management system and be audited to that, be able to address the 21 CFR Part 11 requirements for clinical trials and the GXP requirements for clinical trials, that we would be able to plug DNAnexus as a global hub, an internet accessible, secure, compliant global hub for all the players and actors in this clinical trial space. As I pointed out yesterday, there's clinical trials management, the systems of record, the electronic data capture. There is the web randomization services that are used for partitioning participants in the clinical trial. Obviously, you've got a bioinformatician that needs to develop the pipelines associated with the trial, and the study physicians and the sequencing lab and CROs that are feeding the data up into the clinical trial. The DNAnexus platform in this case can really manage the clinical pipelines in a way that the clinician can only use validated pipelines. It'll also, we've actually had to go and change our organization. I pointed out we have our new VP of Security, Compliance, and Quality, Lauren Buell, who started, and it's mandated under GXP that this function has to be independent from engineering. It cannot be somebody that is on the pivotal path towards delivering a product or a service out. There has to be this level of independence, and so we've actually staffed that up as well. If you were to look at why QMS is required, you really are having greater regulatory scrutiny at this point. The agencies have definitely stepped up their enforcement and the corrective and preventative actions for noncompliance. Authenticity and confidentiality of data is paramount, and this is again where the 21 CFR Part 11 and other parts come in. The auditability is also very important, and this is where the platform features of DNAnexus, beyond just the security and compliance, things like data immutability. If you put a piece of data in DNAnexus onto DNAnexus, you can't change it. You can delete it, but you can't change it. The versioning of applications, where you know exactly what application versions run, what input files are run, what reference sets are used, what input parameters are used, all of that is tracked and logged. You can go back and reproduce any of this, and that's a real key aspect of the clinical trial capability. The flow of information is important. As we saw in this diagram, you really do have a very clean way to use DNAnexus to integrate all these players on a global basis. You could see clinical sites being distributed, and I think that's going to be a very big help. Finding clinical sites for the wide variety of specific cancers and other diseases is going to be a challenge, and you can't just keep going to the same places for a clinical site, and the broader you can cast a net, the more clinical trial participants you can get, the better likelihood you have of a successful trial. The global outsourcing, again, as I said, having an internet-based hub, secure and compliant, enables you to really reach out globally from wherever you're trying to develop this trial. And of course, the security has to be aligned with HIPAA. All of this stuff is protected health information that we're dealing with. The specific quality objectives of our DNAnexus quality management system are all around pipeline consistency and reproducibility, security and availability of the data integrity is maintained at every step, and then our continual improvement, as I mentioned yesterday, is in place to detect issues and to control changes. And again, the continuous improvement is about making sure that if there is a problem in the system, and I'll give you a great example of where quality failed. Invitae announced about a month ago, they're a carrier screening. They announced about a month ago that they found a process flaw. They had to retest 50,000 people. And it actually has an impact on the industry because this is an LDT, and you've got the FDA looking with some scrutiny at lab-developed tests. And to have a lab-developed test where a quality problem allowed 50,000 of these things to go through. They indicated that only 15 false positives or false negatives were there. I can't remember which way it was. But the fact that they had to go and retest 50,000 people, that's not good. Again, that was a full failure of a quality system where they did not have their process fully checked. And another exciting piece of news, what we're working on for the FDA, We have been working Precision FDA for the last 2 years, and it's been very successful, as you saw with groups like Sention, the deep variant folks from Google. It has been very successful in elevating the standards for regulatory science around NGS-based drugs and devices. The next step for the FDA and for Precision FDA is to get to the point where they can actually do review submissions. So this is not going to be the entire 510K review submission process baked into the Precision FDA platform, but the portion of the process referred to as interactive review, and it doesn't even have to be for 510K. There could be other types of approvals and other types of submissions that sponsors provide. But the key is they're going to be able to do this entirely on Precision FDA. So if you think about submission of NGS pipelines, submission of reference materials, the FDA needs to validate. And essentially, the heart of Precision FDA is a VCF comparator. And what they're looking for is if you have a diagnostic or a test that you can get the pipeline, the FDA can validate that the variants that you're seeing that are relevant to your test are consistently produced by your pipeline. So the FDA gets to check your reference materials, the FDA gets to check your actual informatics code. And the way they're doing this is what they call airlocked review spaces, where there's a space for the sponsor, a space for the reviewer, and the reviewer controls access into this shared central area. So the sponsor can work independently, test their pipeline, get their evidence prepared. The reviewer can create templates for reporting, provide specific types of pipeline analysis or comparators that the sponsor must use, and then they all meet together in the shared area. Everything that happens in this space is logged and audited. You can't write a sentence inside this space without having it be tracked. So at the end, you can go back and pull everything out in a full auditable log, and that can be put into the system of record. So this is pretty exciting for us, and this is what's driving our FedRAMP moderate expedition as well, which, as I explained yesterday, is lots of fun. And I think that's my time. Thank you very much.
Wow.